Excellent. Thank you very much for the kind introduction. I would like to talk to you for next 45 minutes, right? About meso and more importantly, macroscopic modeling of the brain. So what I will be talking about is brain network modeling. So let me first introduce what I mean by that. The brain network model is a network derived from connectivity defined from empirical data, whole brain empirical data, where the individual nodes of the network are equipped with some neural mass model. When we solve the equations in time, we get a source activity or the activity of the model in a state space, and that we can transform with the observer models into something which resembles the data which we can measure from the individual people, like fMRI, Bolt, or EG, SEG, or MEC data, and so on and so forth. So let me now go, the actual equation would be this integral differential equation with noise, where we have the individual node dynamics defined by the neural mass. Then we have the, let's say, neural field connectivity, right, which goes over the surface of the gray matter. And then we have this heterogeneous connectivity, which is derived from the white matter tracks. Of course, in this model, we can also introduce external stimulus. You can think of, for example, all things like natural stimuli, like a sensory stimuli, or some invasive or non-invasive stimulation. Don't worry, the slides, I will share it with you. And of course, some noise, which can absorb all sorts of phenomena which we are not directly modeling. So let's go now over the individual components in a little bit more detail. So the node dynamics, as I said, are governed by the neural mass models. So we are not modeling the individual neurons as it was in the previous talks, right? So we are now zooming out on the larger scale. But several different flavors of models have been derived. Mean field models were derived to capture the collective activity of populations of such neurons, right, or neuronal populations. So, for example, here this is the ADEX model, where the activity is captured as a mean firing rate and mean membrane potential of adaptive exponential point neural models, right? And it can be demonstrated that under certain conditions, these approximations hold. What we lose completely is of course, any temporal ordering of these spikes, right? We just get the mean activity. So if you just remember that was yesterday, right? This sort of information we lose. As I said, there are several flavors of these models, so they can be based on firing rates or local field potentials, or they can be completely phenomenological, not tying together to a specific definition of the neurons. So, for example, we can have just some kind of oscillators, or a specific phenomenological models for a specific purpose, like our favorite epileptor model, which represents the local transition between seizure and non-seizure state in epilepsy. I will talk about that later in the next section. Then we have the connectivity, right? Not only the regional one, as I said, which goes between the brain areas, which are defined by a particular parcellation. So this would be the coarsest structure encoded in the brain network model. But we can also have, as I said, the surface-based connectivity. So you can think of it as taking the surface of the brain as a mesh, discretized mesh, right? And then applying some connectivity kernel across the nodes of this mesh, right? So that would give you the surface connectivity. And of course, knowing what the model does in the so-called source space is interesting, but we want to compare this directly to the data we can acquire from the individual subjects. So we have also different observer models, right? So for the different modalities we can apply. So for example, for the EG and CG, we need to compute the so-called gain matrix, right? How the electric field propagates from the sources, here defined by the nodes of the brain network model, and how these then sum to the signals which we observe in the sensor space. And of course, for fMRI, there is a model, for example, the balloon-Dickinson model, which describes how the activity of the neuronal population translates to the hemodynamic through the hemodynamic response function to the Bolt signal. Now, I have been spending quite a lot of time on the connectivity and how important it is. Why am I talking about the individual people? Right. Well, it has been shown in this case in mouse that the individual connectome derived from a particular individual, in this case not the person but a mouse, has a much better predictive power when it comes to predicting some functional observables, in this case functional connectivity, than the group average. And an individual connectome, I mean, derived with the same modality which we can apply also to humans, the diffusion tensor imaging. But of course, it's not. It could be better, right? We all know that the DTI has its own problems coming from the resolution and the inability to distinguish orientation of the fibers and so on and so forth. In mouse, we have the possibility to compare this against tracer data, right? Which is an invasive technique of mapping connections between different parts of the brain. And also here, it can be compared against in this case, it's the functional connectivity, remind you, against the scan-rescan variability, right? So we are here with the individual connectome, so it's not stellar, but it's not that bad. Yeah? So sorry about that, this doesn't start at 0. Oops, that was wrong button. So to sum up the first part, the brain network model has 3 main components. We have the nodes, which are governed by the mean field models. The network has edges derived from a personalized connectome and the connectivity on the surface of the brain, potentially. And we have the observer model, which translates it to useful signals. We can then compare with the empirical measurements. And lastly, the individual connectomes derived from the non-invasive data increase our predictive power. Now, as I said, I spent quite a lot of time on the structure. So let's try to relate this to the function, because that's exactly what we can do with the generative model. So in our hands, the behavior, and now this can be fairly general, is a function, some function of the structure, right? In this case, of the brain. In particular, as I was saying, we have the DTI data and the MRI imaging to supply us with the information of the structure, and then from that we can generate all sorts of functional data and compare it with respect to some functional data features to the empirical world. One way how the structure constrains or can influence the function is through the space-time structure of the connectome. So what do I mean by the space-time? Of course, the space is the network topology, right, of the connections. Like what is connected to what are the hubs, what is the, I don't know, clustering coefficient, and so on and so forth. And that defines or describes, that's how we, with the graph theoretical measures, can describe the structure. But in the brain, what is important is the propagation of the signal across these connections. And these are associated with some distances, and the propagation velocity is finite. So we get time delays, right? We get and very different time delays for short or long connections, and depending on the myelination and so on and so forth. The delays have been also recovered functionally from in vivo data, either in the abstract database from SEG. So this is a group study on the response to stimulation across recorded from the intracranial data, and also from MEG or EG from the resting state. Delays between, let's say, avalanches or their propagation across the brain. And so what we have shown also is that the delays shape synchronization and dynamics. So what do I mean by that is that it is about interactions, right? How the different nodes of the network interact with each other. So these can be either frequency independent, which then the information here travels across the lengths, across the fibers as like buckets of information or some let's say particles. Or it can be also frequency dependent, that the oscillatory activity of the individual nodes then influences how the information is processed or transmitted. Right, so we can have then several cases where either here is just where there is no oscillatory activity and the information just flows as particles, or it does so also, but there are some background oscillations, but this does not influence the information transmission, or if it's the case that the information transmission or the interaction, right, interaction between the nodes is dependent on the phase, for example, of some oscillations. Then this slide and the next one matters the most, because then what the brain is doing, the function depends on, for example, things like on a phase or on a phase coupling between the different nodes. The advantage of the brain network models is that we can set up simplified models, right, which are analytically tractable, and study this phenomenon, focusing just on this, right? So what a colleague of mine, Spás Betkosi, is showing is that if we consider the delays in the network, we get a different distribution of, let's say, hubs, right, communication hubs, depending on the frequency of how we set up the network. And this is really interesting, because that means that all these let's say results from the network approaches to the brain function, which are showing the importance of hubs and the rich club and so on and so forth, which consider the, let's say, particle way of communication within the network, can be refined based on the frequency of interest. And what this is showing is there is an interesting correspondence to what can be recovered also from empirical data, where the, let's say, different functional networks have a different preferential frequency on which they communicate, right? Or on which they, let me correct myself, on which the interaction is preferentially happening. So, that was about oscillations. But though we really like them, because as I said, it's analytically tractable, the brain is not really an oscillator. It's a pattern generation machine or system. And one way how to study these patterns is through functional connectivity dynamics. So as Sasha was mentioning, functional connectivity is a funny name in neuroscience for correlations. But these are usually computed across the whole session, right? And now I'm talking about sessions for resting state experiments. These would be like 20 or 5 to 20 minutes of a person lying in a scanner, not really doing anything. And we can summarize such a session with the functional connectivity, right? Compute the correlations of the bold signals, and we get a general shape of the interactions, or what the functional connectivity is like. But we are completely losing the information of the dynamical properties, right? How the for example, how fast is the activity unwrapping, giving rise to the particular functional connectivity. So we can study that, these dynamical properties we can study with functional connectivity dynamics, which means that we compute the correlations in a sliding window across the whole session, and then we compare mean again correlate these functional connectivity matrices against each other. And that gives us the FCD matrix, which is time by time instead of nodes by nodes in the functional connectivity. And then we can try to describe or quantify some properties of this matrix. So for example, we can try to look for some states, which would be here, noted here as these blocks, right? Which are, you can think of them as intervals where the functional connectivity or the structure of the interactions of the systems is constant. And then it switches to something else. Huh? We can also look at something which we called fluidity. That means how fast this switching between the different functional configurations is happening. And this has been shown to be quite expressive and useful, for example, to track differences across ages or different conditions and so on and so forth, right? So here have a let's say, highly fluid dynamical functional connectivity of a young person, and then more static dynamical functional connectivity of an old person, right? So something like that. So now the question is, if this is the functional data feature or the phenomenon we want to explain, how does this tie to the structure? And one way how to address this is through structured flows on manifolds, where we are showing that through breaking of the symmetry of the system by introducing the connectivity, right? So the symmetrical system would be either fully connected or completely disconnected, whereas the connectivity with the structured connectivity being somewhere in the middle. This lowers the dimensionality of the exhibited dynamics, right? So the disconnected system, of course, is fully symmetric. Every node is doing whatever it wants, and explores fully the available state space. If you introduce the connectivity, this available state space collapses to a much lower dimensional manifold, and then on this manifold, as you move along it, you will get different flows. So we can study this very well in the model, because there we can actually change, in this case, the global coupling, right, to introduce the symmetry or to destroy it, and we can show that there is like a one optimal working point in which the fluidity of the system arises, right? So here these would be the functional connectivity dynamics matrices as they pop out in this working point. Again, remind you to remember the talk of Sasha von Albara from yesterday, when she was showing something similar for the connected point neuron models, right? Where there is this critical, not critical, but important choice of the right coupling strength to get interesting dynamics. The dimensionality here we can measure or observe through embedding the system in with either PCA or some other nonlinear method, showing that here we have in the symmetrical system we have basically just the sphere, and then this takes some sort of shape as we go near the working point, and this shape is then lost again. I was showing here these flows, so that would be the manifold, and the flow around it would be the system going from different parts so for example, from here to here, where here it can be, I don't have here the details, they don't matter that much, but I can tell you that here, this would be the place of the, let's say, low activity of the system, right? And then the system can slowly transition here, where more of the nodes are in a higher activity state. And then exactly here we can observe some highly correlated fluctuations, which then translate to the individual events or these blocks of strong functional connectivity on the FCD matrix. So. Just to summarize this section, the symmetries and the symmetry breaking pose constraints to the system dynamics and lead to dimension reduction in the functional exhibited function. The connectome in our hands provides such a symmetry breaking by its space-time structure. And the low-dimensional subspaces allow us to capture the global complex dynamics such as multi-stability, heteroclinic orbits, or avalanches. And I would add across different timescales because, for example, the multi-stability we observe mainly on a slow timescale in the fMRI or fMRI-like data, whereas the avalanches is a property of a much faster on a faster time scale, usually observed in EEG or MEG and so on. Yeah? And in this model, we have captured both of these. So I promise I'm almost done with the theory, and just a few applications and how to embed this in some sort of inference scheme. So the first application I would like to talk about is the virtual aging brain, where we are trying to capture the inter-individual variability as we, and now we, I mean the individuals, are tracing some trajectories. For example, in the virtual aging brain, it would be the aging trajectory, right? Where as we go with the age, with different brain phenotypes, there are changes in the for example cognitive capabilities, right? How do we do that? Well, we do it with the help of cohorts. So in this case, this is the 1,000 brain study from Jülich. Just not 1,000 in the end, something like 600 subjects, but still, which maps spans the, let's say, later aging time span. So these would be subjects 50 years and onwards. And for each of these, we can build a personalized virtual brain. We can then, for which we can of course then simulate data. But more importantly, in this cohort, we have not only the structure, but also the, again, resting state functional data from which we can derive, both from the structure and the function, some interesting data feature. And with the help of the modeling, we can try to put them together in a coherent framework. Why would that be interesting? Well, not only to, let's say, understand or provide some mechanistic understanding, but more importantly, we can then try to embed this in some inference framework and be able to, for a particular person, be able to estimate the parameters of, and not only of the model, but also where on the aging trajectory a particular person can be placed in a particular time. And here, just a side note. But aging, of course, we can ask the person how old is he, and that's not the point. The point is that the biological age does not necessarily correspond one-to-one to the brain age. Yeah? So, in this case, how we are with the time, I have plenty of time. So in this case, again, we built a whole brain model from the DTI data. Use the same model as before for the SFM, these flows on manifolds. But now the The interesting observation from the structural data was that there is a decrease in interhemispheric connections across the age. Right? So this was something which was robustly found across the cohort. So that's exactly what we have encoded in this parameter. Let's say alpha matrix. So here this is 1 for the interhemispheric connections and 0 for the intrahemispheric connections. And now we can actually, so to say, virtually age a person, right? So we can apply this mask to the empirical connectome with a variable strength, right? That would be the alpha parameter, and observe how the functional data features are changing with that. And that's exactly what we did. The functional data features again would be here the functional connectivity dynamics and its variability, which was also shown to be decreasing over age, right? And we can show that if we apply this mask, so to say, in a virtually aged person, that the functional connectivity dynamics variability or the fluidity also goes down. So, second application I would like to talk about is epilepsy and epileptic surgery. So we're of course not capturing here epilepsy in general. We are focusing on drug-resistant focal epilepsies, right? Which is a fairly several disease, for which the only alternative is then epileptic surgery, which is preceded by exploration of the epileptogenic zone with the help of the intracerebral electrodes. So there are implanted electrodes intracranially in the suspected epileptogenic zone. The patient goes off the drugs, and then the recordings of the seizures are supposed to help the surgeons or the epileptologist to identify precisely the epileptogenic zone and then mark it for resection. This is a fairly old procedure, like from 50s. It's done until today with few or little changes, and unfortunately has only something about 60% success rate. So there's definitely a room for improvement. It's exactly what we are trying to do. And why do we hope to be able to contribute something is because in this case we have a network data feature, right? We have the propagation of the seizure across the network or across the brain. We have a network fitting target, as in data fitting target, that would be the epileptogenic zone, right? So that's a label you can assign to several nodes of the network. And there is a way for also network-based intervention. So there are possible interventions would be then either the resection or reconnection, disconnection, and so on and so forth. In particular, the model which we apply here would be the epileptor, which I already mentioned at the beginning, which is a phenomenological neural mass model which reproduces the temporal dynamics of the seizure. So that would mean that you have some interelectal activity. It's like the normal resting state. And then there is a characteristic transition into this highly synchronized hyperactive state, which then after a while transitions back into the normal state or interelectal state. The epileptor model has these 5 state variables. The first 2 governing the fast discharges, so that would be this activity. Then there is a very important slow metabolic process, which basically governs the probability of transitioning between the interelectal and the ectal state. And the other two state variables govern the, let's say, finer data features such as spikes and waves. The parameter x0 here, that's this one. That's the most important one, because it controls the excitability, or you can also think of it as a epileptogenicity of the model. If it's lower than a certain threshold, the node by itself will never transition spontaneously into the ictal state. Whereas if it's over that threshold, this transition can happen just without any outputs, any inputs. Again, as before, this is a fairly simple model, so we can study it systematically and analytically, or with the help of some numerical methods. And for example, here, Marissa was showing that with this model, you can basically put together a taxonomy of possible seizure types, right? Coming out of the bifurcation structure of these equations, which then can be found also in the clinical recordings. But that's just a side note. What we actually want to do with this model is to use it to estimate the epileptogenic zone, right? So how do we do that? Again, we take the empirical recordings. We derive a data feature of choice. So in this case, that would be, for example, power in the fast frequencies, which captures well the transition between the interictal to the ictal state and back. We take the epileptor model, which or a simplified epileptor model, which can again perform this transition between the resting state activity and the fast discharges, right? So this would be the resting state and the fast discharges. And then we combine these two in a parameter estimation framework. So this could be in our hands. These are Bayesian inference schemes based either on a variational inference or some sampling algorithms such as Hamiltonian Monte Carlo, right? Where the empirical data are used to build the brain network model and provide also the empirical data features, and then this goes in a loop and finds the parameters which if we apply them to the model, provide the best fit. And it's not only about the best fit, and then resulting, of course, into some clinical report, where for each of the nodes of the network, we have an estimation of the epileptogenicity and everything which is above some threshold, we can mark for the clinicians as potentially epileptogenic. Going back, I said that it fits the data and provides you the parameters. That's actually not the point. I mean, we can do that, but we will discard a lot of information because, as was mentioned in the previous talk, degeneracy is a feature inherent to not only the biological system, but also to our models. So many combinations, potentially many combinations of these parameters can lead to the same observables or the same data features, functional data features. And this is something which, of course, if you apply an optimization algorithm, you don't get, because you get just the point estimates, right? So that's why we are opting for the Bayesian approach, and we are trying to provide distributions of these parameters at the end. Yes? So you have the possibility to assess the variance or the how certain you can be about a certain estimation. This here is now running as a part of a clinical trial. One of the first applying, or I think the first applying brain modeling to epilepsy. This is happening currently in France in 12 hospitals. So, to sum up, I showed you that the whole brain modeling has the capacity to explain functional variability within and across individuals by exploiting the structural variability and provide causal hypothesis about provide and test causal hypothesis about brain mechanisms. And a second, that the capacity to personalize the brain network models have been shown to have a high predictive value. So, to conclude, brain network modeling and in respect the numerical implementation in the virtual brain allows us to close the gap between the models across scales and brain imaging. And second, I would say that the dynamical system viewpoint allows for mechanistic understandings of brain function and dysfunction. And lastly, the personalized in silico brain modeling performs, the platforms enable us to search for novel clinical solutions. So with that, I would like to thank you for your attention, and I welcome any questions.
Thank you very much. So we've got first question.
Hello, thank you so much for your very elaborative presentation. My question is, I wonder if there are any other clinical trials for personalized brain modeling for other neurological disorders, because I can certainly see the value of such modeling for progressive neurodegenerative disorders or even demyelinating conditions. So that's the first question.
Yeah, I think that's an excellent question. I'm not aware of any, though so we are definitely not at the stage of a clinical trial, but demyelination or neurodegenerative disease is of course a highly relevant or a very good potential application of these approaches. So I was talking about the virtual aging brain, right? That tracks the trajectory across the healthy aging. I mean, that's interesting, but more interesting are the deviations of these healthy trajectories, right? Where you have, let's say, some diseased aging or non-healthy aging, and currently we are also working in parallel on a very similar project, again on a cohort level for Alzheimer's disease, right? So there would be the NDD version of that.
Okay, thank you. So the second short question is whether there is any trial to associate this modeling in conjunction with diagnostic tools, say, use this modeling in conjunction with diagnostic tools, say fMRI for a particular disease. So rather to shift the probable application towards diagnostic rather than assessing the severity.
I see. Yes. Yeah, in the context of Alzheimer, that would be definitely as a diagnostic tool, not as a potential treatment, because there the issue is that for Alzheimer's we don't really have an intervention available. The only thing which we can do right now is to try to diagnose as early as possible. So of course, any tool which provides you improvement in predictive power of this diagnosis would be helpful. So that's why brain network modeling would be a good fit here.
And one last very short question.
In your original brain network model, I would like to know if you have intensively studied the phase transition based on noise. Different kind of phase transition in the dynamics.
And you mean like taking different flavors of noise, or
a different state of noise based on noise, where we will lose the functional connection in lower dimension.
So if you drive a system with noise, right, you can turn a system which just have a couple of fixed points towards it would converge into a system which actually exhibits very interesting and fluid dynamics, right? So one of the systems I was presenting has a two fixed points. Is the low activity or down state and the up state. The down state is a stable it's just a fixed point, right? Whereas the up state is a spiral. If you consider this just deterministically without noise, even if you initialize close to the up state, it will just converge and stay there. If you drive it with noise, you get these, like, let's say, pulsating oscillations, and of course transitions between the up and down state.
So if we can predict based on this kind of analysis different kind of dysfunction, even before we compared it with fMRI data, that was my.
That's interesting. I would have to think a bit more about that, but we can discuss offline.
Yeah, sure.
Thank you very much for a very interesting talk.